Именно tos. Мне составляется. Մեջները որ լոգոյի մոտ լնես։
Աստորեն դուք Վլադիմիր Գյորգիևիչ
Լավ շպրտեցիք ենք։
Հիշեք, Վլադիմիր Գյորգիևիչ, հենց տենց ենք ասում։
Սաղովինը կանստեք, մենք ետ ու երեք։
Մի հատ էլ առանց դիմակի։ Այնքը աննայք քուրն է, այնքը չեմ ասում։
Вот, есть же указка. Вот она, видите?
Уважаемые коллеги, я надеюсь, уже все собрались, самые стойкие участники нашей завершающей сессии сегодняшнего дня. У нас сессия посвящена вызовам современной науки пандемии COVID-19. Я рада всех приветствовать на нашей сессии и рада представить первого докладчика, профессора, доктора медицинских наук Наталью Юрьевну с докладом о полученных уроках и задачах на будущее в COVID-19.
Уважаемые коллеги, добрый день. Большое спасибо, что пришли на нашу сессию. И позвольте мне начать мою презентацию. Попрошу технических ведущих вывести ее на экран. И моя презентация, основная задача была это дать таким широким мазком то понимание, к которому мы пришли в итоге спустя год после начала пандемии. Поэтому я так и назвала «Полученные уроки и задачи на будущее». Куда мы сейчас движемся? Если посмотреть на те математические модели, которые пытались дать прогноз ситуации в ее развитии на перспективу, то, например, одна из таких моделей, которая вот у меня на среднем из представленных картинок вверху, она наиболее все-таки, так скажем, приблизительно, относительно точно отражает ту ситуацию, которую мы сейчас наблюдаем в развитии в Российской Федерации. Я думаю, это не зря, потому что именно в нашей стране все-таки все ограничительные меры были приняты заблаговременно, объемы тестирования, старт производства, они все именно послужили именно той причиной, что развитие ситуации в Российской Федерации хоть как-то было прогнозируемо и даже вот соответствует одной из математических моделей. И если посмотреть на то, как развивается ситуация в мире, то, конечно, здесь много разных непредвиденных факторов, появление новых штаммов, в том числе, которые начали циркулировать в разных странах мира обладают большей инфекциозностью. Но, тем не менее, даже если соответствовать тем прогнозам, которые были даны на этой математической.
And now we are in the middle of that criteria, and within 1 year or 6 months, we will have a complicated situation in some countries. A lot depends on the infectious control measures taken in different countries of the world. And I would like to remind you the situation that we had in Russia in the beginning of the pandemic, when there was a large-scale testing launched in Russia for all of the people which were arriving from abroad, and there were some measures taken, and the test systems were developed at a high level, and also there was a production of new diagnostic test systems in February and March. Several platforms were launched to manufacture the vaccines, and we managed to stop the infection spread, and we achieved the peak last May, only 2.5 incubation periods after the registration of the first 1,000 cases, and it helps to decrease the load for healthcare system, which was only training staff, and was launching production of personal protection means in a larger scale than before, and we solved that situation, and that was not so bad like in other countries. Yes, that was very difficult, but we had the abilities to solve that, and we could manage that situation, which we had in winter and autumn this year. So the virus continues to mutate and be spread along the planet, and there are different strains which are more infectious, from UK and South Africa, and the Brazilian strain as well, which has high transmission rates. That's why we are limited in our air transport with the Tanzania and Turkey, because these strains can be imported, and this is a serious problem for all of the provinces of Turkey. They already found the British strains at 52 provinces. They are the red zone. How can severity of COVID-19 may change over the next year? The mathematical modeling that I would like to present to you. So this is a scientific study from our international colleagues, and so this according to seasonal coronavirus immunity. So the immunity which decreases the severity is a long-term one. After endemic phase of the infection, after the primary action with the new coronavirus infection was in the children's age, our prospects are quite good, and the coronavirus will be seasonal and will not cause such severe diseases in adults, because the contact with the infection will be happening at children's age, at young age. And also the contact should be with genetic determinants which cause production of antibodies. We should all be vaccinated despite the risk factors and the age, so that it becomes a seasonal infection, and also Um, we know that in children it doesn't have such severe progression. Children can be infected with all of them, and in teenagers, all of them, they have the ABs. In adult age, when you face the seasonal coronaviruses, so you do not have symptoms or the symptoms are very mild, and that's different for elderly people, but in epidemiological process, the children's populations are important. You see that the reproduction index may achieve up to 6. Non-specific prevention measures prove their efficacy, decreasing by dozens of times the probability of infection, social distancing, and protection of the eye mucous layer, and also the protection of other mucous areas. And yeah, we should stick to that. There are lots of publications about that. And I liked one of these publications. It was presented last year globally, and it is demonstrated that in the state of Tennessee, when there was no requirement to wear masks, there were such areas there. The prevalence compared to summer increased by 200%. They also the patients with respiratory symptoms, and where the population stick to all of their recommendations, practically there was no increase in incidence. Social distances and masks and gloves, they really work. And we should also think about flu vaccination, and the mortality among elderly people with COVID was lower if they had vaccine against flu. They were not having flu. Practically, we have no cases of flu now, but there were important steps towards this in many countries, those who had vaccination against flu. And we should think about pneumococcus infection as well, so to vaccinate against that. And most study the pathogenesis of the disease, and I would like to remind you what is the peculiarity. It is related to late term immune response and production of interferon type 1, and interferon type 1 launches the pro-inflammatory cytokine synthesis, which activate the macrophages, and the cell immunity is activated, but it's all happening at late stages. And the immune response is extracellular, and it's not related to the virus itself. It is related to pathological hyperactive immune response, intracellular response, which provides for protection of the cells, and which provides for apoptosis, which can be induced with interferon type 1, and non-cellular, all of these things which have influence on the other parts of the immune system. And also non-infectious pneumonia, it in these cases was provoked by coronavirus. Another thing we should remember is that the severity of the disease is determined by genetic factors. And we see people with the risk factors who do not have severe progression. Yes, there are some genes which regulate the immune response and the factors of immune response and severity. And also innate errors in TLR3 and IRF7 dependent production implication of IFN first type. So these are the inborn errors which are responsible for life-threatening progression, and it is involved with interferon 1 activity. And autoantibodies against anti-interferon can be found in 15% of patients with a critical condition, and most of them are males and people who do not have severe progression, no symptoms, they don't have antibodies against interferon. There are ongoing discussions for a long time that interferon type 1 is the main component of inborn innate immunity, and they experiment for SARS-CoV-1 and 2. IFN delayed production is related to lung damage and mortality. The early introduction of interferon 1 can prevent the lung damage. The use of recombinant interferon to stimulate the antiviral host response is also tested. Now there are clinical trials ongoing. And in the chart to the left, you may see that in this case, the patient interferon production achieved its maximum on the 5th day of the disease, but that was late. The patient has already the critical condition. And pathogenesis of COVID-19 and flu. So we can see that in the slide, because the obesity is a risk factor for severe progression of both flu and COVID-19. So the secret is adipose tissue, which produces, in response to inflammation, adipokines, which are pro-inflammatory cytokines, and the TNF alpha and IL-6, they were found in adipose tissues. Yes, there are pro-inflammatory cytokines, but they cannot perform this function. And the pro-inflammatory cytokines, they are at high level as a response to inflammation, and there is activation of macrophages, and they start to damage endothelium of vessels. And as some of the people, especially with diabetes mellitus, who were getting or are getting chemotherapy, for example, related to some cancer pathology, it causes more dysfunction of endothelium, and we find thrombosis in such patients. CNS damage, so there were some doubts about that, and we started to understand that loss of smell has a connection to CNS system, and it is related to the olfactory nerve cells, to their damage. And another factor is related hypoxia. They are related to vessel damage, which causes thrombosis and ischemic insult and ischemic stroke. And also all of the receptors are used by coronavirus, and also there is leakage retention in one part of the blood vessels and deficit in the other, because angiotensin 2 is ACE2 is in too high qualities, and the central nervous system is also suffering because of hypoxia, and also there is encephalopathy, encephalitis related to direct impact of the virus. And the search for drugs started a long time ago in China. They tested all of the available drugs, and you may see that Arbidol was also tested, and that was among those drugs which were listed, and it had a different name, and beta B, maybe what was analgesic, or what we know is used for flu. And some of these drugs are still used, and some of them demonstrated no clinical efficacy as of today, but therapeutic targets can be different. So this is the antiretroviral drugs, which block reproduction inside the cells, and they prevent the virus antigen inside the cell at the stage of interaction with the receptors. And those drugs which impact the consequences of coronavirus, we are talking about blocking the pro-inflammatory cytokines and so on, and also the medicines which may find coronavirus before contact with the cell. I am talking about monoclonal antibodies or anti-COVID convalescent plasma, which is registered for clinical trials, hyperimmune globulin. And yes, we have prospects. What we understood as of today, the treatment should be distributed in time, because many patients they did not timely receive adequate medical care, because the doctors did not know how important it was to distribute prescription of these drugs in time, that they should not prescribe A, Bs, and hormones on the first day, that we should wait for the indications, and antiviral therapy on the second week is not so efficient, because the virus already did what it had to. Nevertheless, so we were the pioneers here, all of the healthcare, all of the countries, to get a more clear understanding what we could use for these patients. Sometimes, vice versa, we conclude that some of the drugs are inefficient. For example, one of the recent recommendations by WHO on drugs, guidelines on drugs for COVID-19, according to them, you should not prescribe to patients hydroxychloroquine and lopinavir, ritonavir, and we do not recommend that. And even at hospital stage, remdesivir did not prove its efficacy. Maybe it should be done before days 5 and 7, and also for the corticosteroids. The experts of WHO, they said yes, they proved the efficacy. It makes sense to prescribe them, but in proper dosages. And let us remember about flu, to extrapolate that data to COVID. So the antiviral therapy, it works only in the first days. Prevention and treatment of thrombosis, China did not say anything about that. Probably they had no such cases. So Europe was the first to face such situation. Maybe people were more senior there. And I learned about that when I was on humanitarian mission of Russia. I visited Italy in that humanitarian mission, and the doctor said, yes, we have a problem. Many people, they die from thrombosis, and also these recommendations, they became well known to all of the countries, and Italy started to actively use inhibitors of pro-inflammatory cytokines and hormonal drugs, because the first recommendations were we should not use hormones because this is a viral infection, and those recommendations were taken from flu guidelines, and unfortunately it turned out to be opposite. The hormones work, but not from the first day. And as for steroids, so we should be more careful about them. According to WHO and other countries, they say that it's enough to use therapeutic dosages, not the pulse therapy, because pulse therapy may lead to increased Vladimir levels, which speaks about the fact that such patients have higher thrombotic risks. Moderate steroids should be the basis for hormonal treatment here. And also know that why first they were furious about these drugs, because they may foster sepsis and secondary infections. but when we need them, we should use them because they are lifesavers. Post-COVID syndrome, many people are talking about that. So up to 3 months, this is the long COVID. So after 3 months, up to 6 months. So this is what we understand post-COVID. And for right clinical manifestations of this disease, we are talking about the patients who are PCR negative, starting from long-term oxygen dependency, ending with the CNS damage, and which may be related to oxygen dependency. It may be also for mild forms, and there is a term brain fog when you have memory issues.
Мысли. И, наверное, тоже некоторые, те, кто перенес
And those who were just ill, who were sick, so they also face that. And thrombosis, which are related to a long-term hypercoagulation, and for a long time after the patient is PCR negative and he's released home, he should be taking anticoagulants. So of course we have lots of learnings, and SARS-CoV-2 is more infectious compared to flu, and pathogenesis is different from flu, and and retroviral drugs, they with the proved efficacy, so they are inexistent, and it can be assessed only for patients when they are used very early in time, and they should be prescribed in the timing when they may be efficient, and the basis of treatment should be well-regulated pathogenetic therapy, because unfortunately there are no antiviral drugs. Non-specific prophylaxis, they decrease infectious risk and the virus mutates quickly. Tasks for the future: continue to improve the algorithm of treatment, study new drugs for prevention and treatment of SARS-CoV-19, increase the volumes of tests, control the virus expansion, and continue to stick to all of the non-specific prevention measures before we are all protected, vaccinated. Thank you for your attention.
Спасибо большое, Наталья Юрьевна, за такой широко информативный доклад, за ответы на большинство вопросов.
I think there are quite many questions that are relevant to all of us, especially those who faced the infection or could be infected in future. Thank you very much for such relevant information. Dear colleagues, the next report will be delivered by Elena Gavrilova. She will be talking about a comprehensive response to SARS-CoV-2 virus. Good afternoon, dear colleagues. Can I have my slides on screen? First of all, I would like Today I would like to talk about comprehensive response to SARS-CoV-2, and I today introduce State Center of Rospotrebnadzor, that is a leading virusology center in the Russian Federation, and facing such a challenge, scientific challenge, we had an opportunity to use all the extensive experience we have been accumulating for more than 40 years fighting various viruses, and in January 2020 we developed diagnostic tools to detect genetic material in clinical samples. 2 PCR kits I'm talking about. First, they were the first kits registered in Russia, and also if a kit in order to detect antibodies in clinical samples. These kits were produced, and as of today, we have produced more than 18 million kits. They are distributed to all regions of Russia, and we also provided more than 1.5 billion testing kits overseas. In January 2020, we got a status of reference lab of COVID-19, and that allowed us to do clinical diagnostics of all clinical samples that we got from all Russian regions. As a reference laboratory, we developed protocols for tests. We detected resilience of genetic materials in various transportation media in order to detect most sensitive methods. And also we detected the most representative material for clinical studies. Today we do understand that that would be respiratory swabs, but back in 2020 and January 2020, we certainly didn't have that information. So we studied genetic material in order to understand better the virus. Besides, starting from June 2020, in order to detect mutations, we have been studying the new coronavirus. Every month we get samples from all over Russia. We do genetic sequencing in order to detect modifications in the virus. It's a very important work in order to track the modifications in the virus, and also to understand the efficacy of the used materials and medications. So we have, as you know, there have been detected a number of modifications of the virus, even though significant changes have not been found yet. The most important, though, is carrying out virusology studies. So when we got a virus from Australia in February 2020, we started to carry out a number of trials, including animal studies, culture studies, and so on. And based on our own transgenic cell lines, we started to divide more. We divided more than 1,000 Russian isolates from different parts of Russia. After getting the virus last January, we started to test it on various animals in order to use them in further trials. So we have defined that there are 3 groups of animals. The most sensitive turned to be ferrets and monkeys got infected with a much higher virus burden. So we have defined markers together with the swab tests. Additionally, so these works have been carried out for quite a long time. Therefore we have developed transgenic mice that express a certain marker, and this transgenic line of mice can be infected with coronavirus with high titer in lungs and in the brain of such mice. a separate series of studies have been done in order to detect comorbidities. It was especially relevant before the start of flu season. We did a number of studies to when we infected sensitive animals with coronavirus, and a very important conclusion we did was that the infection so infecting coronavirus after getting the flu infection can lead to very serious complications. So that's what we see in the groups of animals. In comparison with mono infection, the situation was much better in the mono group. Apart from, it was very important to hold trials on how to save the virus at various media to understand what disinfecting agents was it sensitive to. We did a number of trials in that regard that showed that the virus can be sensitive up to 48 hours on most surfaces. At the same time, it is sensitive to disinfecting agents, a wide range of them, and it's not sustainable in chlorine water. We have performed a large screening of various therapeutical methods together with pharma companies, scientific institutes, and so on. We studied more than 800 combinations of therapeutical agents on culture cells, and we also checked prospective medications in vivo on the animals. As for the vaccine and medicines that are used today, according to WHO data, more than 250 medications are being tested today. In our country, there have been registered 3 vaccines. And starting from spring of 2019, Vektor, using a multi-platform approach, have developed 6 prototypes of vaccines against the new coronavirus. And here I would like to draw our attention to the most prospective vaccine, Epivac-Corona. It's a peptide vaccine. There have been studied 15 different peptides while developing this vaccine on sensitive animals. The most immunogenic peptide have been chosen, and a combination of 3 peptides together with the carrier protein, which is nuclear protein. We got a vaccine that gives a very high immune response among sensitive animals. Then we did a full range complex of preclinical studies. It was conducted on 3 groups of animals that showed a very high immune response. Antibodies titers were very high. As for protection, among the monkeys, we did vaccination of the said animals, and saw 100% of protection. In placebo group, in comparison, 67% of animals had two-sided pneumonia. As for safety of the vaccine, we did a 3-stage study.
And as we had to quickly do the tests, And after the first stage, we received the approval for clinical trials, and the second, third stage, we did that in parallel before registration was done. We started the trial on July 27. That was the first phase with 100 volunteers split into 2 groups. And in these clinical trials, we demonstrate high safety of the vaccine. There were side effects only, pain in the place of injection, and the vaccine in 100% of volunteers on day 42 caused production of specific antibodies and virus neutralizing antibodies. As of clinical trials of the first stage, the vaccine was registered, and we shifted to post-registration trials for 65 plus, and the second trial was expanded for 3,000 volunteers. So this is placebo-controlled, double-blind, and so we had these trials going on in 5 regions, and for in 94% of volunteers it induced specific ABs, in 77% virus neutralizing antibodies. Based on the results of these clinical trials, and it was allowed to be used without any age limitations to all of people or with the age over 18. And if we come back to the comprehensive approach of fighting against COVID-19, when in December we all started to hear about the British strain, thanks to tracking people who came from the UK, thanks to centralized system of Rospotrebnadzor, we managed to quickly get the clinical samples and separate the virus, and also its virulency and resistance to prevention medicines and antivirals. So we demonstrated that in the serum of immunized by Evipigvac and Sputnik V patients. So they neutralize the British strain together with the reference strain, which we get in the beginning of the of the band. There is no high virulity using the animal models. They are detected by all of the diagnostics kits used in our country. So, and so this type of works are done at top international level, and it also gives us an opportunity to do a complete cycle from fundamental studies of the new virus to development of diagnostic preventative medicines, their clinical, preclinical trials, and launch of production. And this is a huge experience that we all received over the last year, and we also gained experience now. That was a serious challenge for modern science. Thank you for your attention. Tatiana, Elena, thank you for your interesting presentation. I would like to thank all of employees of Vektor. So you did a huge work, which is so important for us, and I was witnessing how you were starting. I can say with whole responsibility that the merit of a better epidemiological situation in our country belongs to employees of Vektor Institute. Thank you very much. Thank you for such kind words. I would like to make the presentation, and I would like to present the presentation of Vladimir Dedkov. He will speak about WHO mission in China and how they saw the origins of coronavirus there. So, dear colleagues, in fact, well, this is not actually my presentation. And this is the presentation that I'm making on behalf of the group of international experts which were studying the origins of SARS-CoV-2. I would like to just explain, well, around those missions there was a lot of policy, and the situation with the COVID-19 is kind of too political, and I think this is and should not be like that, and I would like to explain about this mission. And of course, epidemiological investigations 1 year after something happened, well, somewhere, well, those who deal with the quick response methodology, you understand that if there was an outbreak in a canteen and you come in a week's time, there is nothing you can find there. If it was after 1 year, of course, we cannot be talking about some urgent responses. That was not a political or policy mission, and that they were experts of WHO, and there was no political investigation, that we were just investigating, and the Chinese, they were just explaining, proving that they are right. That was a purely scientific mission, and that was a united mission. On one hand, there were WHO experts. On the other hand, there were Chinese scientists. And the goal of the mission was to summarize the experimental knowledge and the results of trials and tests, study some other information from other countries. That was accumulation of all of the experience available, and based on that cumulative analysis, the hypotheses were made on the origin. I would like to emphasize these are hypotheses and they will be tested. This is only the first part of the big work which will continue, I hope. And this is only the preparation. You may see here the expert group. It included independent experts from different countries. To provide for maximum transparency, and WHO representatives were involved, and also Food and Agriculture Organization, representative of the World Organization for Animal Health, and 17 from Chinese colleagues, and that was a joint study by WHO and Chinese experts. What were our goals? And the data analysis, retrospective analysis, and visit of the key sites which were related to spread of the virus, and based on that, planning out the strategy for phase 2 studies. And I did not translate the slides into Russian on purpose, and they have the original view so that you understand how it all works. And there are epidemiological studies. They were trying to understand whether the virus appeared before it was detected. They were trying to detect the very first case because nobody could understand whether the first person found that was the first sick person ever, and the COVID-19 may have different types of clinical manifestations. It can be asymptomatic and severe respiratory syndrome with different complications. It could have circulated before, but nevertheless. So this is the genetic trial sequences of patients who got sick in December and January, and comparison with the molecular epidemiological data and comparison against biological general data. And also this is the study of the animal world, animal studies, studies of markets of food and supply chain and the cold chain, and the study of virus circulation in the environment in potential hosts. And then all of these data was summarized, and we were divided into 3 groups, and what we worked for each area, we summarized all of that, and which gave us one interesting report. I can say that we visited certain sites which were related with the onset of the pandemic. That was the legendary Huanan market, seafood market, Huanan. There are the hospitals for the first patients, and other wholesale markets to see how the sale works there. And that's where the labs of the regional CDC, municipal CDC in China. This is like Rospotrebnadzor, surveillance body. That is the Chinese Rospotrebnadzor. And we also visited the Institute of Wuhan, the Institute of Virology, and many are thinking that it's because of them, they are the cause of the pandemic. We also spoke to people in the municipal regions who were sick, who faced the disease, so they shared their impressions. We analyzed that. We just try to figure out and understand when the pandemic started. We know that the first cases proved by lab tests in humans, and it was proved by genetic tests. The virus was found, that was on December 10. So probably the virus was circulating before, and we were trying to look for signs of virus circulation, and it had an impact on the circulation, and our Chinese colleagues did not notice that, and we could not find such signs retrospectively our Chinese colleagues, they started the medical records of 76 plus cases, and which took place in December, January 2019-2020, and 92 cases were attributed to COVID, and 1 year time after we took samples from 62 patients. All of them turned out to be seronegative among the people who got sick with a flu-like infection. 9 were positive from the number of tests in January. What was surprising? So we analyzed the prevalence of flu-like infections, and there are no special changes before the pandemic, what we could see. And moreover, if we look at the long-term changes, we can see that before that situation, there were no typical changes compared to previous years. And the time of onset of the pandemic was the end of November, beginning of December. The same we can say if we analyze the mortality, and it's clear when the pandemic started, so the mortality started to grow. And not all of the deaths were due to COVID, and with the onset of the pandemic, so it's added extra mortality rates. If we look at mortality increase from pneumonia, so this is the not hospital pneumonia, not in the hospital, in Hubei province, then what we can see there, so this is what we can see, this is the market. And this is how it was spreading. And in terms of the time, so it is in line with the timing, December, January. Looks like the pandemic started when we imagine. And what was surprising, those who heard the news, so you heard that there was some animal on the market, and in fact that was not really true, as we may see from the slide. More or less half, 40% of the cases of the disease in December, they had no link to human market epidemiologically. And I also witnessed conversations with the people who were sick in December, and they said, yes, we are not related to that market, we never went there, and what should we do? Just and yes, actually these people were in another district that's not some special market where all of the city goes to buy something. This is just a municipal market in one of the districts, and it's interesting only for those people who live nearby. And people do not just travel there on purpose from other parts of the city, and there is a question mark still left. And the market turned out to be the place for mass infection of people, and after that, that was impossible to just get rid of that. And from one place, people were hospitalized in severe condition. And of course, and I know what about China. Well, that doesn't look like good sanitary conditions, but it was clean. There was the final disinfection, and the sanitary condition was not good, and any infection could spread. That could be any digestive tract infection. So all of the conditions are present for that purpose. And well, this is the point of sale and WC nearby, so the toilet. So it looked like that. Lots of people, bad sanitary conditions, no surprise. It's not that, well, the pathogen had all of the chances to widely spread here to provide for mass infection. According to one of the versions, that version is offered by the Chinese when the virus was brought to the market with frozen goods. As a proof, they mention certain samples with the virus, not only using molecular methods, but also it was seeded, it was cultured, the real life strains coming from frozen food. And the problem is that the samples were taken in the second half of January, when there was the pandemic already running, and probably this is secondary contamination by sick people of those foods. And these foods are different, they have different origins, and you can see that the 460 different frozen foods from 9 provinces and 59 countries. That's very hard to imagine that at the same time all of these products can travel from all of these countries. At the same time, all of them are infected and to the same just provincial market in China. We did not have that in other places. Well, actually, if we looked at the plan of the market and what was sold on the market, well, the market is called seafood market, but in fact it was not like that, because on top of the seafood aquatic product there was meat, and also there were wild animals and pets, and you understand that the pets are also snakes and crocodiles grown at special farms. They're used as food, and they use in traditional Chinese medicine for different purposes. And well, we're kind of not really used to that, and it is all located in a way that here we have all of the sale points for these types of product. It's not only seafood, it's poultry as well, and meat of wild animals, and so you know, we cannot find one source, but it's disseminated in the western part, in the western area of the market, and it's split into two areas, two big areas, and there is a corridor between them, and mainly all of the cases of the disease which we had, they were found in the western part of the market. In the east, that was not really happening. If we look at the types of products where the viruses were found, in which points of sale, those who were selling poultry, who were selling the seafood. So more or less that was like in the western area meat, the food was different, but it was in the same place. So if we look at the percentages in which foods and how often you can find this infection, so seafood was the most frequent case here, and the rest of the products or the rest of foods are here. For example, in the poultry, it's not less. I think so there won't be significant difference between those percent. There were lots of infected products, and I don't think this speaks about some one source of infection. On the other hand, using molecular methods, we analyzed the early sequences from mid-November, early December. We used TMRCA, and the first place, these methods, they provide for assessment of the time when the virus appeared. According to estimates, it is in mid-November, beginning of December. Well, here I should mention that those methods are accurate. The more sequences you have for analysis, and the sequences can attribute to January, November, early December. They are not so multiple. It's about 70 of them. Good quality sequences that we could work with. So this amount of sequences does not provide for a good result, but it is like that mid-November, early December, which is in line with epidemiological data. And so we can see that those sequences which were received from samples from the market, they are quite genetically even.
Различают.
So genetically they differ. In other words, they do not have a common predecessor that they would go apart. So that points to the fact there could be a number of infection sources. At the same time, we do not understand what could be the source of the infection. It could be a sick person that infected others, or it could be animal origin. So we try to study a number of animals, including bats. So there are no bats that live permanently on the markets. It is located in a vivid area that usually are not favored by bats. As for rodents, yes, there are some evidence of rodents, such as feces. Apart from that, so trying to find a nature origin of the virus, our Chinese colleagues did significant work. They studied samples of bats, samples taken from pets, including molecular methods, serology methods, and other animals they could get samples from. As you see, they have taken a big number of samples from 24 provinces, but we got no results. No traces of virus have been found so far. So there are quite many questions, but as for results, we have today we created 4 hypotheses. First one, so first 2 theories are linked to a natural origin of the virus. The only difference between the 2 is the spillover was direct from some animal, or there could be spillover through an intermediary. It could be either this or that way, but all experts said that most likely is that the virus had natural origin. As for the versions of cold chain, it is not as likely as the previous ones, because it's not clear how products that come from different countries could arrive to a market, infected products I'm talking about. So it is not supported by sufficient evidence. Well, at least we have to study it further and speculate a theory of laboratory leak is quite unlikely. We still take it into account, but there is a very low chance that that was the start of the virus spread. The most likely source of coronavirus is a bat. Why bat? Why not? Why not another animal? As I said, as of today, nobody have found SARS-CoV-2 virus in bats. At the same time, bats at different times show viruses that are very close to the said coronavirus. So they are different, but they are like siblings, if you could say that about the viruses. And at this phylogenetic tree, these viruses are shown. And virus RATG13 is mentioned as a prototype of virus SARS-CoV-2. In fact, we still do not have any serious proof that this was the reason, but still there is a theory that is very popular among average population. So another evidence that points to bat origin is as follows. If we look at the coronaviruses that are pathogenic for human beings, include virus SARS of the first type, MERS, and so on. And as you see, bat is seen in each virus. So I'm talking about NL36 virus, 63 virus, and so on. So, and the transition of the virus is from bat to human beings, sometimes through a camel, sometimes through rodents, but at least there was a preliminary animal, so which proves that spillover was not direct, but it was through another species. So such development is quite likely. Why not? Yes. Our previous experience points to this fact. So this slide I took from Petr Daszak, who for quite many years have been studied various coronavirus varieties in nature, starting from the detection of the first coronavirus, SARS. From our own experience, we know that a variety of coronaviruses is really very wide, not only in bats, but in other animals as well. All animal groups show at least one or two coronaviruses. If we take a gut sample, we regularly observe it in various trials. We have just not drew our attention to that fact. In fact, there are some people that have been dealing with this problem for quite a long time, and for them this story is no surprise. As for Institute of Virology in the city of Wuhan, well, here the cause and effect sequence has been changed because the lab that works with the viruses in this institute got its specialization because they wanted to study varieties of coronaviruses in nature in order to predict outbreaks of such viruses. And they started to focus on it after the first outbreaks of the first SARS virus. So the original goal was to prevent the spread of such viruses, but now they're being accused of doing it on purpose, doing the spread on purpose. At the same time, there is no proof that shows the leak was from that lab. We were in the lab, we examined it. It's in line with the current standards. The institute undergoes accreditation annually with WHO specialists. At this institute, all operations are in line with the requirements. Of course, we cannot claim that last year was everything was okay, but today at least the operations are absolutely adequate. Moreover, if you look at this map, the red pointer shows the location of the institute, and the yellow one the said market. This distance is quite big, and apart from that, this territory is divided by the river of Yangtze. It's not as wide as Dnieper, but it's a it's a big river. And as for clusterization of COVID-19 that took place in December of 2019, it is against the wind that flows. At least if there was a leak from institutes, from the said institute, we would see some clusterization towards the market, but no evidence of that have been detected. So today all experts believe that lab leak is a very extremely unlikely origin of the virus. Well, that's it for my presentation. This is our joint team of Russian and Chinese research group. If you want to get more detailed information on the investigation materials, please use this link and get the report that provides details of the investigation. Thank you very much. Well, thank you very much, Vladimir, for such a very interesting and detailed report on investigation. I believe that retrospective study is very important because that gives us all tools to be well prepared for any situations in future. So this was extremely important. Thank you for your fantastic work. And I would like to give floor to Tatiana Gribennikova, who will be talking about approaches to develop recombinant vaccine against COVID-19 for HIV-infected patients. Good afternoon, dear colleagues. First of all, I would like to say thank you to organizing committee of the conference for invitation, and I would be talking about approaches to develop recombinant vaccine for HIV-infected patients. Pandemic that was announced in March 19 changed the whole world. Today, it is not over, as we all know. And we continue studies, scientific studies. So if we talk about HIV patients and how COVID-19 influenced their life, a number of studies were conducted in that regards. Previously, the studies conducted in 2020 show that 80% of HIV infected were men with the average age of 53 years, and all of them had antivirus medications. As for comorbidities, they included hypertension, diabetes, obesity, and other infectious infectious diseases such as HB and HE. The most common symptoms were fever and cough. As for severity of the disease, it was either moderate or mild. So the first reports suggested that HIV patients do not have a higher risk of being infected with COVID-19. And moreover, the patients that have antiviral therapy have less chances to get infected with COVID. Among the dead patients, 90% of them were older than 50 years old, and most of them were men with a number of comorbidities. In 2020, such studies continued. And as for immune status and severity of disease, there are a few theories. First hypothesis is that a low immune status can protect human body from cytogenic storm. At the same time, analysis of other scientists shows that there is a correlation between immune suppression and severity of disease. Fortunately, it did not influence much the outcome of the disease. Further studies show that multimorbidity and old age influence the severity of disease and death from COVID-19. So, hypothesis shows that antiretroviral therapy can help to protect against COVID-19, not only among HIV patients, but among other patients as well. Such medications like lopinavir and ritonavir were checked against COVID-19 and initially were used for the treatment of COVID-19, even though the efficacy was not proved further. Tenavir showed inhibitor activity, efficacy, and showed a better result in trials, but today it's clear that standard antiretroviral therapy does not protect patients that live with HIV. We can see that people living with HIV can be infected with COVID-19 and affected by risk factors and disease progressions. Multimorbidity and old age are also among risk factors. Despite a number of observations that antiretrovirus medications can improve the patient's conditions, it is clear today that standard antiviral therapy doesn't protect patients from COVID-19. For people living with HIV, an effective way to protect against COVID is certainly vaccination. Probability of complications is much lower compared to consequences of COVID-19. British Associations of AIDS divided a number of patients that are recommended to be vaccinated. First of all, it's with people with the level of CD4 lower than 50 to 200 cells per milliliters. Those who had opportunistic disease in the last 6 months. And also patients with CD4 levels at 50 to 20 cells per milliliters, but together with immunosuppressive therapy, patients with comorbidities with CD4 levels from 50 to 200 cells per milliliter. With COVID-19 and the HIV infections, a number of immunologic parameters change. CD4 cells regulate B and T cells. A volume goes down when the number of H cells goes up. The number of CD4 lymphocytes with acute infections among HIV patients goes down, especially at early stages of disease. The influence of the virus at immune system leads to the life of CD4, and there is a redistribution of these cells between blood flow and the lymph nodes, which contributes to lymphopenia. Therefore, in the period of immune deficit, it could lead to the higher permeability of gut, of digestion tract, and other systems. So what shall be the vaccine for people, for HIV patients? First of all, it should not contain a live virus or any other genetic material that can replicate. It shall be effective against the main strains of SARS-CoV-2 and activate various type of immune response. It should be safe, free of microorganisms, and oncogenic. All these requirements are included in the recombinant vaccine based on virus similar particles, virus-like particles. They cannot replicate, therefore it's absolutely safe. VLP have a large number of virus sequences that that act as virus similar epitopes. They can develop the immune response in a very short time, and they can also activate the immune response of the saliva. As for the selection of the protein that shape VLV is done through monitoring and methods of molecular epidemiology. Phylogenetic research help to detect the viruses. As we all know, the virus has been mutating starting from the outbreak of pandemic. There are new glides and new mutations that can influence to detection of the virus and the efficacy of vaccinations. Therefore, analysis of full genomes in Russia and the neighboring countries is extremely essential. It makes it possible to modify the content of the vaccine, and mutations that activate recombinants make it possible to include at the first stage and therefore they influence the expression. As for specificity of the antigens can be checked by immunoenzyme analysis. We see that VLP interact only with serums that are positive in neutralization reaction. Moreover, the signal of compare the result of with dilution 1 to 320 and 1 to 8C. Immunization with VLP helps to develop a significant immune response But as for antigen, it's not the only thing we have to do. The next step is to optimize the virus and further trials. Therefore, as co-author of this report, I included a number of our staff in place that did this complex work. We hope to develop a recombinant vaccine that is in line with the highest requirements, which will allow us to vaccinate HIV patients. At the end of my presentation, I wanted to say the following. In Russia, there have been registered 3 vaccines that make it possible to prevent the spread of this very complicated vaccine. But it's very likely to see more vaccines on the market. The biggest learnings we got in this pandemic is that in such a complicated situation, the victory can be done only with the use of comprehensive approach. And the fact that we have 3 registered vaccines and the fact that we have been developing new vaccines shows that potential of the Russian science is not limited. Therefore, I would like to say that only with the help of the efforts of the international scientific community, we can overcome the virus. Thank you very much for your attention. Thank you very much, Tatiana. And making the conclusion of our session, I would like to say that Russian science provided an adequate response to the COVID pandemic. It helped to address a number of issues. Certainly, we will have to answer lots of questions in future. We have launched a number of centralized programs aimed to understand better the virus, its mutations, and those programs are carried out both nationally and internationally. Certainly there are a number of challenges that we will have to address, but the overall work of the scientific community will certainly help us to achieve success and solve all the problems we've been facing. Thank you very much for participation in this section. Thank you, dear colleagues.